Thank you very much for inviting me. It is a privilege to address this very distinguished audience. I represent not just the vaccines theme, but also the larger cluster in infection and immunity. And I guess I'm the more pleased to be here, Dame Fiona, as I represent a minority group in the vaccines theme, simply by the genetic defect of having a Y chromosome. We are about 300 researchers in vaccines in Oxford broadly, the large majority of whom are female. So we have made progress. I'm going to talk about vaccines, which I guess are they are a British invention, which has been rather impactful. So the estimates from WHO these days are that vaccines save around about 2.5 million lives each year. It's almost certainly the most cost-effective medical intervention ever deployed, and disease eradication efforts are very much on the cards again after smallpox, with polio nearly there and measles coming along. But more broadly, vaccines are being used and will increasingly be used in a whole variety of age groups for a very large number of indications. We already have human papillomavirus vaccination for teenagers, TB sometimes used in that age group, meningitis increasingly, and then for older adults, of course, flu vaccination, but a very effective shingles vaccine coming along, and several others. So I'm going to talk to you today a little bit about the work of the theme in those important areas, but also on new target diseases that I think are very much on the horizon, as diverse as vaccination against psoriasis, trying to cure hepatitis B and C with therapeutic vaccinations, and arguably the final frontier for vaccination, which is cancer. Keith mentioned that it's important to have infrastructure. It is crucial. We're very lucky in having a purpose-built centre for clinical trials, the Centre for Clinical Vaccinology at the Churchill Hospital, which rather luckily is outside the door of the only GMP manufacturing facility based in a university for vaccines in Europe. Purpose-built clinical trial centre and the clinical biomanufacturing facility. And we are, as many of you know, the only BRC with a vaccines theme. And this is crucial if you're going to do translation, and we are all about translation. Of course, we use animal models, but we move as quickly as we can into human trials. We've had graduate students who, within the time period of their DPhil, within 3 years, have made a vaccine. It's gone forward into GMP manufacture at this facility. It's gone into clinical trials, and it's shown high-level efficacy in humans. Try doing that with a small molecule. We make a lot of vaccines, to be fair. This is our chart of progress in a whole variety of disease areas. There are 30 principal investigators at the Jenner Institute. Malaria is the largest program, 24 individual constructs in clinical trials. The leading TB program from Helen McShane, who's here this evening, also Ellie Barnes, her hepatitis C program, HIV, flu, meningitis, RSV. I'll focus on a few of these, but not all of them. But I'll start with emerging diseases, which hit the headlines with Ebola just a couple of years ago. Pandemic flu has always been a threat. We were careless, really, about most of the others. Ebola, now Zika, Middle East respiratory syndrome, Rift Valley fever. The reality is that despite Ebola, we remain unprotected against these epidemic threats. Ebola was a wake-up call. People have sprung into action, but you don't make vaccines overnight. Progress is being made. Our theme had already responded very rapidly through Andrew Pollard's efforts in the Oxford Vaccine Group to the swine flu epidemic in 2009, and the Department of Health were very involved with that and impressed by our efforts. But notably, in the Ebola crisis back at the end of 2014, only 5 vaccines went to West Africa during the period of that outbreak, and 4 of them were tested first in human beings in Oxford in this vaccines theme. You know the story, an unexpected, very large outbreak in 3 countries in West Africa, growing out of control in late 2014, threatening to spread right across the world. And we were called up in about the middle of August by the Wellcome Trust, the Department of Health, WHO, asking could we take a vaccine into clinical trials. It had been made, but hadn't been tested yet. By the end of the year, we had tested several vaccines. We got the first vaccine into a human being in the middle of September, in about 4 weeks. And in case some of you don't do translation, let me just explain this doesn't usually happen. You don't write a grant application to any of our funders, get it peer-reviewed by 6 reviewers in this case, answer the peer review comments, get a decision from the funding agency, and receive the award within 2 weeks. It would be nice if that happened, but it, of course, doesn't. You then have to fill the vaccine, persuade the lawyers to sign long pieces of paper, and actually start the clinical trial. So that was a record, I think, for anyone. 3 other new vaccines reached clinic in Oxford by the end of the year, and efficacy data on one of the Ebola vaccines was published in The Lancet in summer of 2015. So that's 10 months from the first immunization of a human to reporting an efficacy trial. That hasn't happened either. So you can do things more quickly. People are trying to improve the problem or the situation that's summarized on this slide. These are outbreak pathogens, mainly viruses, one bacterium, that could appear again tomorrow and cause very serious outbreaks. We have no licensed human vaccine for any of these. There is a large fund driven by the Wellcome Trust and others trying to raise money to do this. They've been pretty successful, and it's called CEPI. We are partnered with Janssen in making major applications to CEPI to help out with that in partnership with the WHO. And this is progressing really well. In fact, in the lab, we've made candidate vaccines for all of those based on a leading technology that we have developed in Oxford, chimpanzee adenovirus immunization, and that is going very well. But let me turn now to meningitis vaccines very briefly. You know that we now have vaccines for essentially all the main types of meningitis. Meningococcus B was the last, and a new vaccine appeared from Novartis, then GSK, in 2014 for babies. Fairly high efficacy. And the problem was, as people saw, that we were only giving it to a certain age group. And there was, prior to Brexit, the largest petition to Parliament ever, 820,000 signatures, to get that vaccine used more widely. But it's not a perfect vaccine. You need multiple doses. You have limited but strain coverage. And certainly the durability of protection is quite short. So what has happened in Oxford is that Christine Rollier, working with Andrew Pollard in the Oxford Vaccine Group, has developed, using the same technology used for Ebola, an adenoviral vector, a single-dose regime, with remarkably durable antibody responses, so it may well just be a single dose preclinically. And they have taken that vector into GMP manufacture here at the Churchill Hospital. And this is the phase 1 clinical trial, first in humans, that will start in the next couple of months, comparing head-to-head this new vaccine against Bexsero to ask the question, can we devise a single dose vaccination that will be useful for prevention of meningitis? Very briefly, there are, of course, many other major vaccine programs in Oxford. I've listed just 4 of the larger ones on this slide. Malaria has been going a while. TB, the only program to test a subunit vaccine for efficacy in Africa, is the Oxford program, or anywhere in the world. HIV, a leading program initially devised by Andrew McMichael, continued by Thomas Hankey, and certainly the most progressed in late phase 2 universal influenza vaccine. One number on that slide tells the story: 125 clinical trials so far. These have not been easy programs, but real progress is being made. So in our last malaria vaccine trial, we're up to 82% efficacy. So I think we'll get there. One of the ways that facilitates that is to do real experimental medicine, controlled human microbial infections. You may not be aware that somewhere in the world today, 18 different pathogens are being given by people to other people to test vaccines with full ethical approval and regulatory oversight and so on. We've done a lot of this on malaria in Oxford, also influenza. And Andrew Pollard has been so energetic in testing typhoid vaccines that I understand in the latest documentation of the distribution of typhoid in the world, Headington features as a real hotspot. Finally, therapeutic vaccines. Psoriasis. How do you vaccinate against an autoimmune skin disease? Well, if you know that a monoclonal antibody to IL-17 alpha is pretty effective in severe psoriasis, you might try vaccinating against that cytokine, which is exactly what Martin Bachmann's group is doing. HPV, a very exciting program by Lucy Dorrell, still in the preclinical phase, but looking very promising for preventing early stage cervical cancer. How late the stage will be or how late you could vaccinate therapeutically is unclear. Hepatitis C and hepatitis B therapeutic programs being led by Ellie Barnes, who is here. And in cancer, of course, there's been not just an explosion of interest and enthusiasm, but real efficacy. So as many of you know, in the last few years, immunotherapy of cancer has come to the fore. You can take monoclonal antibodies that are checkpoint inhibitors in that they relieve or unblock the immunosuppression locally in a tumor, allowing naturally evoked or induced immunity to work. And these drugs, these monoclonals given intravenously, work on their own to an extent. There was the first one, not very effective on its own. There's a better one, anti-PD-1. We now know if you combine these two, it can get above 50%. And that's the body's natural immune response to the tumor kicking in when you unblock immunosuppression. What if you added a vaccine to that? And that, of course, is what many groups are now trying to do. This is a trial in Oxford. Chief investigator is Freddie Hamdi, that we devised using, again, vectored vaccine technology to induce strong cellular immunity, looking at early stage prostate cancer, intermediate risk patients, vaccinating with imaging before and after vaccination and before surgery, using the biomarker of PSA to look at impact. Over 30 people have now been vaccinated, and we're about to add in a checkpoint inhibitor into this trial to improve efficacy. Similarly, cervical cancer, Lucy Dorrell's program again, efficacy has already been seen by other groups targeting very early stage CIN, in other words, CIN1-2, cervical intraepithelial neoplasia, with fairly modest immune responses. Better vaccines could certainly do better, but could they go all the way to treating cervical cancer? We don't know, but that question is very much being asked. And on my final slide, just a reminder, as Keith mentioned, that we're interested in economic development as well through stimulating the economy through spin-out companies. The vaccines theme alone has leveraged over 50 million in the last 5 years. We raise a lot of money from different funders in several continents, and we're very grateful for that. And we've started 2 spin-out companies in the last year, one targeting universal flu vaccines, cancer, and other diseases, and one a virus-like particle company that will be announced tomorrow. So in closing, I will thank all of our funders and you for your attention.